Okay. So we are going to give you a variety of different information that we've been collecting about black sea bass over the last few years, go into sort of the background and why we're asking the questions that we're asking, give you a little bit of an update on sort of what we know right now about black sea bass in the Southeast region, then give you a little update about where we stand with a coast-wide genetic analysis for the entire Atlantic coast of the United States, and then finish up with describing some new methods that we tried out for capturing juvenile black sea bass. So I would suspect that everybody's relatively familiar with this concept of climate change driving some shifting ranges in a wide variety of different types of species, especially fish species and marine fish species. Black sea bass is definitely no exception along the Atlantic coast. If you take a look here, this is distribution of black sea bass basically from the Chesapeake Bay northward. And you can see starting in their data set in 1974, heavily concentrated in sort of the middle Atlantic. And as we step forward in time, many are moving into that, more northern region. Until today, we regularly see black sea bass in the Gulf of Maine. So along with that, folks in the region have seen, increases in catches in black sea bass, particularly in the last decade, 15 years or so. So concomitantly, what is going on in our region? This is the estimated abundance of black sea bass from the latest stock assessment that just came out. And as you can see, a significant decline in the total abundance, estimated abundance by age, and especially big declines in those young fish. So we here at the Southeast Reef Fish Survey use 3 vessels to monitor reef fish all along the entire South Atlantic coast from North Carolina to Florida, the Palmetto, the RV Palmetto, RV Savannah, and the NOAA ship Pisces. From all 3 of those vessels, we are deploying these chevron traps on known or suspected hard bottom areas, and a frequent resident of those traps, a frequent guest in those traps, I guess, is black sea bass. Important to note that these traps, the mesh size essentially excludes fish, black sea bass, less than about 150 millimeters. And so we're basically capturing the segment of the population that is also available to fishers on the water. Our survey does a good job of capturing black sea bass where they are. We definitely are going deep enough to find them. They are seen throughout our entire sampling range. And so we're pretty confident in the data that we've collected over the past several decades on these populations. So we were able to take this large data set and sort of parse it down a little bit to give us a picture of what's happening at that age 2 time period. And so we took data from black sea bass and assigned all of them an age based on their otolith age or an age length distribution when we didn't have any otoliths available for some of the fish. And then we assigned all those black sea bass to a birth year based on the year of capture and their age, selecting only age 2 fish and analyzing year classes from 2003 to 2022. We used a VAST model, which gives you not only indices of abundance or indices of year class strength in this particular case, but also a geographic center of distribution. Combined with that, we created an annual regional bottom temperature, taking all of the bottom temperature data that we'd collected along with our traps and our trawls throughout this entire region to look at sort of how the overall picture of bottom temperature in the region had changed in the ocean, and then correlating those temperatures, abundances, geographic centers of distributions to one another. So what did we find? First of all, with total abundance, what you can see is a similar pattern to what we had seen in the estimate from the stock assessment. The SERFS index follows the trends of landings, and we see, you know, a pretty big increase in the early 2000s, the mid 2006, 2007 time period, and then steep declines ever since, essentially. When we're looking at the total distribution and the center of distribution, if we zoom out and look at our entire sampling range, the center of distribution is sort of right near the center of our sampling range. And along that coast of South Carolina to Georgia. If we zoom in, we can see in this particular figure, the size of the dot corresponds to sort of the relative abundance in that year. So we see that a lot of times the larger dots are actually farther to the south and the smaller dots are farther to the north. Taking this with our information about each year and stepping forward, we can see that that pattern mostly holds true as well. If we take a couple of example years, 2006, we have the regional bottom temperature is on the top. So the year prior had just been very, very cold. There was an upwelling event that had happened. And although that year's temperature did increase, we then though see sort of continuing to increase in total abundance and the center of distribution moving farthest to the south for our entire time series. Another example year here, we've got 2018. Things are kind of heating up in the region. Our stocks are definitely declined, and our center of distribution is all the way farthest to the north in this case. So these are our observations. This is what we found, but that of course leads us to more questions about why these things are happening. So it could be that these fish are settling in cooler waters farther to the north, maybe making a little move around Cape Hatteras and moving up into that more northern region. Or it could be that these fish are not actually surviving to recruit to the fishery in this particular region. And so Kevin's going to come and talk a little bit more about how we're investigating that question.
All right, thanks for the handoff, Julie. So to test these hypotheses, we are turning towards genetics. Currently, it's assumed that there are 3 distinct genetic stocks of black sea bass, one in the Gulf with a break at the Florida Keys, and then one in the South and Northeast Atlantic with a break at Cape Hatteras. However, this is based on a study by Roy et al., and this was back in 2012. And so as we know, we've seen a lot of oceanographic change since then. And so it's time for an upgrade. And so since we're seeing evidence of a northern shift in the South Atlantic stock, we want to know, are they kind of pushing up and compressing against that break at Hatteras, or have they actually moved north and the break is broken? And so this is kind of what things would look like if the break still existed. We have kind of a southern stock in yellow, a northern stock in blue. So maybe they're pushing up past or right to the break at Hatteras. But if there is a true northern shift, then we'll see those fish actually going past that break. And then potentially even, we're looking to see if there's even mixing between the 2 stocks. Traditionally, there's a little bit of an offset in the spawning season between these 2 regions. So we don't know for sure if we'll actually see that mixing or not. So over the past couple of years, we have collaborated with Virginia Institute of Marine Science with the NEMAP, Northeast Area, sorry, there's always so many acronyms, and I used to work with them. The Northeast Area Monitoring and Assessment Program and the Chesapeake Bay Multi-Species Monitoring and Assessment Program, or CHESMAP. And these guys both have bottom trawl surveys that catch black sea bass. And then we also worked with the NOAA Fisheries Northeast Bottom Trawl Survey. And so you can see we collected samples all the way from the Gulf of Maine down through down to Cape Canaveral. So we have coverage across the whole eastern seaboard where black sea bass are occurring. And so from these samples, we wanted to prioritize so that we had even coverage across the region with about 300 fish from each of the 3 regions. And then we also wanted to make sure we were just looking at the younger fish. So we cut it off at about 300 millimeters so that the results reflect just the current dynamics. So from those samples, DNA has been isolated from the fin clips, and then 13 individual genetic markers have been amplified by PCR. And then finally, PCR fragments were analyzed for length and genotyped using capillary gel electrophoresis. And in this image here, you're kind of seeing what we see from that electrophoresis. So this is one individual fish looking at 4 of those genetic markers in the 4 different colored dyes there. And then the bars are the alleles that we see at those markers. So you have a heterozygous allele in black, a homozygous in green and blue, and then another heterozygous allele in red. And so we haven't done the analyses yet, but this is what we're planning to look at. So we're going to look at population structure throughout the entire collection range using some traditional genetic analyses such as Hardy-Weinberg equilibrium, pairwise FST statistics, and then structure hierarchical clustering. And what that should tell us is, among other things, how many distinct populations we're seeing, where those population boundaries might be, how strong the gene flow is, either between or within those populations. And we can also look at how these boundaries have changed over time over the last decade compared to the Roy paper. And so if we end up seeing distinct subpopulations, then we'll also be looking at molecular diversity indices that will let us, such as observed and expected heterozygosity, inbreeding coefficients, and allelic richness. And these will tell us about the overall population size and genetic health. And it'll also let us know if there's bottlenecks and potential crashes in the populations. And so we don't have the results just yet. We're really close. I think probably about a month or so away. We're still just going through that last Gulf of Maine subset of samples and then a little bit of quality control. But we're hoping to be able to provide these results just within about a month or so. And then I'll pass it back on to Tracy here.
Thanks, Kevin. All right. So I want to go back to something that Julie mentioned earlier. She mentioned 2 potential hypotheses, whether or not these fish are moving north, or also that there's something happening with the recruitment where there's just not a lot of replacement in the southern population or the southern stock. So I want to switch gears to another project that we're currently wrapping up. And as Julie mentioned, when she mentioned about southern recruitment, there's been an observed broadening of the distribution of the northern stock. The southern stock has been declining in the last decade, and now we've documented a shift to the north in the southern stock when either temperature is high or the population size is low. And Kevin's doing more work to establish what that sort of northern movement looks like and the consequences for the genetic stock structure. But the question really remains of whether or not this is from the southern stock moving out of the region, or if it's something like recruitment that has been severely depressed in some areas of the region and effectively concentrates the southern stock into smaller and smaller areas. So I do want to take a step back and define recruitment because the term can be used in different ways. So here we have the black sea bass life cycle, where a fertilized embryo hatches into a free-swimming larva, which metamorphoses into a small juvenile black sea bass that matures into an adult capable of reproducing the next generation of embryos. And during this life cycle, there are 2 typical stages of recruitment. The first being from a pelagic larva into a juvenile that generally occupies similar habitats as the adult and looks pretty much like a miniature of the adult. And this is generally thought of as recruitment to the population. And the second sort of recruitment stage in a life cycle is once they reach a size that can interact with or be retained by a fishery, and that's recruitment to the fishery. And this is often and generally after they've reached a size to allow at least some reproduction prior to retention by a fishery. So as Julie mentioned, the surf traps catch age 2+ black sea bass extremely well. There are very common residents in those traps or visitors in those traps. And so those traps are a much better indicator of what's coming into the fishery, but a direct measurement of recruitment to the population can serve as an early warning sign of what's to come if we have that information available. So we do have modeled recruitment to the population from the most recent assessments. And from the estimated recruitment, they've indicated that we had a very long period of stronger average recruitment to the population up until the late 2000s. But since then, we've entered a period where the models support really weak recruitment to the population. And so, you know, more and more evidence is stacking up that there is an issue here. So if the trap survey can index fish coming into the fishery, but not directly recruitment to the population, do we have a source of direct information about the younger fish? So besides the surf trap efforts in the region, we also conduct the coastal trawl survey, which samples along the coast with a 75-foot small mesh trawl. And you can see that the footprint of the coastal trawl survey now added to the map that Julie showed earlier with the reef fish. So we sample from about Cape Canaveral, Florida to Cape Hatteras, and the gray boxes indicate places where we didn't see black sea bass, but the purple boxes indicate that we actually see a fair number of black sea bass in this survey, with those purple dots ranging throughout the survey footprint. So maybe this is a good option. And as it turns out, the majority of black sea bass collected by the trawl survey are in fact age 0 fish. So they're smaller than that mesh size that's retained by the chevron traps. So this might be a complementary survey for this species. So we asked the question of whether or not we could use this survey to directly estimate recruitment to the population for this species. I appreciate Julie going through the methods. So I'm basically going to say ditto for what she did earlier. We use pretty much the same methods, but in this case, we used only the small fish presumed to be age 0, and the year sampled actually served as their year class. So that way we had a direct comparison from the trawl survey time series to the chevron trap time series. But we did use the same types of models to create an index of abundance and describe the distribution. I am only going to focus on the index of abundance today because it was definitely a good news, bad news situation. So the good news was that we were able to create a time series of age 0 black sea bass from the trawl survey, but the bad news is that when we overlay it with the existing data from the surf chevron traps, they don't match up at all. If this was a good sort of complementary survey for black sea bass, you would see sort of peaks and valleys in those same years, and they should line up perfectly. However, we really don't see that. We do know that the trap survey is a good, robust data set for older black sea bass. So we have no reason to sort of question that as not being sort of like the thing that we should be looking to compare to. But in contrast, the trawls don't catch anywhere near as many black sea bass, and the catches are pretty sporadic. And since the trawl survey occurs mostly on soft sediment, this probably is not prime real estate for juvenile black sea bass. And this is primarily because they just don't have the things that they might need to survive, in particular shelter and places to hide from predators. And so at this point, we don't consider the trawl survey as appropriate as a direct estimate of recruitment to the population. All right, so do we have, maybe there's another way, maybe we have some other things that we could do. In 2022 and 2023, we had some funding to try other ways to sample for juvenile black sea bass, as well as some other species. And so we took on a new project to sample from the estuary to the mid-shelf using small mesh traps and small hooks to beaky rig standardized hook and line. And we sample between northern Florida and southern North Carolina for this effort. So I want to start with whether or not we actually collected juvenile black sea bass as sort of our starting point, whether or not this even worked or was feasible to do. So shown here are the link frequency plots for our experimental small traps and our small hook and line. The gray bars on these graphs indicate the percent of the fish that occurred in any given length bin in centimeters. And the red line that's vertically about midway across the graph are the size at which about 50% of the black sea bass females are mature. So this can give you sort of a visual on whether or not, based on size, a fish is likely to be a juvenile, i.e. reproductively immature, or an adult and reproductively mature. So black sea bass caught in the small traps and the small hook and line ranged from about 8 centimeters or just over 3 inches to almost 40 centimeters or almost 16 inches. So neither gear was terribly selective for just juveniles, but they did in fact collect them. So it is sort of feasible to find a couple of gears that would work. All right, so based on identifying a couple of gears that can collect juvenile black sea bass, we have some suggestions for what a future direct black sea bass recruitment to the population survey might look like. So on these maps, the circles show places where we collected black sea bass, and the X's are places that we didn't in both years of sampling. We collected them from the estuary to the mid-shelf and from Florida to North Carolina, which supports including a variety of areas and states. And this next graph shows the relationship between the abundance of black sea bass in both gears on the vertical axis and the depth sampled on the horizontal axis, with the samples available for analysis shown as these tick marks along the horizontal x-axis. The average abundance is the black line, and the variability around the average is the gray shading. So in terms of the depth sampled, we found that there was a peak in abundance around 15 meters, and there was fairly low variability around that abundance in that sort of shallower range as well. Whereas the variability got much higher the deeper we went. So this really suggests focusing on shallower depths for the survey compared to something like the full chevron trap survey that goes out to 100 meters. The next couple of graphs will have abundance again on that vertical axis, and the thing that we tested on the horizontal axis, for example, this one is gear, but this time the gray bars are the average and the black crossbars are the variability around the average. So for the two types of gear that we looked at, the small hook and line and the small trap, we did tend to catch more black sea bass in the hook and line than the small traps, but you could or might need to use both hook and line and traps for juvenile black sea bass. They do both work. Additionally, we also sampled areas of very bottom relief from no relief at all, our flat sediment, soft sediment pavements, to high vertical relief or hard bottoms, and found that you may need to sample a variety of vertical reliefs as well, with a little bit of a tendency towards more black sea bass in the harder areas with a little bit more relief than others. And then finally, we sampled 3 different habitat types: artificial reefs, natural reefs, and soft bottom. And the survey would really need to include artificial reefs and natural hard bottom more so than soft bottom, with the greater abundances of black sea bass in the 2 hard bottom areas than soft. And this also helps to support our concern about using the trawl survey as a recruitment index, because it is also so focused on soft sediment. All right, so where are we at so far, and what have we kind of learned from these 3 projects? Well, the black sea bass distribution within the South Atlantic, based on our chevron trap survey, contracted north as the population size decreased and the temperature increased. This might be a shift north, or it might be a compression of their distribution at that genetic break. And so we'll really see that by looking at that genetic structure along the US East Coast based on the samples that we collected in 2024, and we're almost done with analyzing. Chevron traps estimate recruitment to the fishery pretty well, but we don't have recruitment to the population currently from any long-term surveys in the area. Recruitment to the population really needs all South Atlantic states sampled, estuaries and offshore sampled, and several types of habitat to be sampled. And with that, we'll be happy to take any questions, but I do want to acknowledge our various funding sources for all these projects: MARMAP, CMAP, MARFIN, the council, and our sampling partners, CFIS, ChessMap, NEMAP, and the Northeast Bottom Trawl Survey. This really could not be done without all of these people pitching in and helping. So I really appreciate everybody's efforts. Thank you.
Thank you, Tracy, Julie, and Kevin. That was a great presentation. I'm trying to take back control if you give me just a second. All right, and just a reminder on how to operate the webinar. Once again, you can enter questions into the question box right here. You can raise your hand by clicking on the hand icon, and then we'll unmute you. You just need to click on this red microphone, make sure it turns green. That'll indicate you're able to speak. And with that, turn on my camera as well, so Tracy's not the only one.
I'm putting
All right.
Julie in the camera.
So with that, are there any questions from folks online? While people are raising their hands or typing questions in the question box, I'll ask a question for Kevin, since he's the one that's not on the screen. There he is. You had mentioned that there's pretty distinct spawning differences between the northern stock and the southern stock. Can you describe those differences that you're aware of?
Yep, essentially the northern stock, the season has shifted a little bit, but there's an offshore inshore movement with those with that population as well. So in the southeast they spawn more in the wintertime, and in the northern region it's more spring fall, I believe.
Which is warmer time of the year. So if it's based on temperature, there's the potential for mixing or for, overlap in spawning season. I don't know. We'll see.
Yeah, so if are the water temperatures about the same when they spawn?
I don't know for sure. I can look into that though.
Just wondering if aquaculture has looked into some of these differences between the two, if there's different growth patterns between the two different stocks.
There is quite a bit of information from an aquaculture perspective on the northern stock, but we have not been able to find any information sort of specific to the southern stock in that realm.
Trying to move all these things around. So Judd Curtis has his hand raised. Let me unmute you, Judd, and then I'll go to a question online. Go ahead, Judd.
Hey, y'all. Nice presentation. I was curious about the genetics. I know it's still preliminary, haven't shown any results, but the marker panel that y'all were investigating, so like 13 or 14 different markers or something, are all those neutral genetic markers, or are there some that under selection? And if you could, if there's any differences between, I guess, if you have any preliminary results between those two, that might give any indication on what the population structure looks like, and then also any factors that might be contributing to the differences by looking at the differences in the genetic markers.
These are all 13 markers are microsatellites, so they're short tandem repeats. Essentially, a relatively fast turnover rate, so pretty commonly used for population genetics studies. As far as is the question about differences that we're going to see within our study or with previous studies?
The main question is, what the marker types were. So the microsats, typically neutral markers. So it should be comparable to the other studies. So I'll be interested to see what you'll find.
All right, this question is going to put you all on the spot. Which of the two hypotheses on the movement of black sea bass seems to be supported right now, if any?
Oh, I don't know. I mean, based on our survey data, there is something going on at the southern end of the survey where we just aren't seeing the black sea bass that we used to. And we certainly saw the full complement of size ranges back in the day, which would kind of indicate that maybe there are no new babies coming into that area. And so that's effectively creating a truncation down there. But there's also no reason not to think that they're not following a temperature envelope as the area has gotten warmer. You know, last spring and summer, we had temperatures above 27, 28 degrees Celsius regularly. And that's unusual. That's really unusual off of Florida. You know, whereas there should be more upwelling down there typically. Other thoughts?
Yeah, I don't know that we can actually answer that question about, you know, sort of what's happening at the northern end at this point. I think that's still very much an open question. Obviously, you know, when we're thinking about it, sort of our vision stops at Cape Hatteras. So it's very difficult to have any idea what might be going on beyond that.
Yeah, and you guys did a recent trap survey north of Cape Hatteras, right? Looking at potential habitat shifts in the two areas. Can you describe some of that? And then we'll go to the others that have questions.
Yeah, so sort of immediately right around the corner from where the survey historically has ended at Hatteras, there is some very, very comparable bottom type that occurs in the rest of North Carolina. And we saw the sort of normal complement of fish there too, like sea bass, vermilion snapper, red porgy, et cetera, triggerfish. North of that though, on the suspected hard bottom was either fairly different from what I've heard, and that was mostly the Pisces that did that work last year. It just wasn't quite the same habitat type, and primarily the catches were sea robins. So, you know, there seems to be a habitat break in that area, as well as then a break where sort of our species stop, or at least for what, you know, we were sort of able to identify from last year. Great area for sea robins, though.
Thank you. Jack Cox has his hand raised. Go ahead, Jack.
Yes, sir. Can you hear me?
Yes, we can.
Okay. Yeah, my name is Jack Cox. I'm a snapper grouper AP member, and I'm a veteran sea bass pop fisherman up here in the Morehead City area of North Carolina. My question is that I guess it's been about 5 years ago, maybe a little longer. I don't even know. I can't keep up with the time anymore, but there was some extensive work with NC State with some tagging with Tom Burgess. You remember that? And I was just wondering if any of that information is showing that these fish are moving further north. It seems like, you know, a couple of years after that tagging research was done, a lot of those fish are showing to have stayed in the area and didn't migrate very much. Can you guys elaborate on that, please?
Yeah, that's what I remember too from a few studies that were done around that time. Jeff Buckle, Paul Ruttershausen, I think Nate Batchelor was involved in some of it too. And they really were pretty site fidelic. They weren't moving very much once they kind of hit that adult stage. So if that kind of holds true, that once they sort of settle into a reef, it really is more likely that any sort of, you know, migration northward movement is probably happening in earlier stages or from a sort of larval delivery rather than an actual adult migration. That's mostly what I know about tagging.
Okay, thank you for that. And in some of this information that you guys got, was there any fishermen involved in helping with the study as you guys were doing your sampling?
So they've been involved sort of supplementary to the reef fish survey design. We've gotten lots and lots of suggested points to build up our universe over time. That's been one of our primary sources of information on where to find hard bottom. For the fin clips, we had actually targeted using the fishery independent surveys because we wanted to focus on getting those smaller, younger fish. So we had a more sort of recent snapshot of what was happening with the genetic structure.
Okay, thank you.
All right, now bouncing to some questions online or some comments. Amy Duke said, thank you for a great presentation. Tracy, I appreciate the distinction between recruitment to the population and recruitment to the fishery. That's something we should incorporate into our conversations.
I'm a larval person from back in the day, so.
And then Haley Scott had a question. If I prepared a comment on behalf of several captains who this would be the time to make it, or there will be a time for comments later or questions. Haley, if you'd like to go ahead and make a comment, you're more than welcome to make a comment. This is not necessarily a public comment session, but there are council members that are listening. So if you want to say anything aloud, please raise your hand. Go ahead. All right, we'll come back to you, Haley. So Lauren Gentry had a question for you all. Do the two stocks have enough differences in their shape or color that experienced folks on the water would have a feeling or for whether or not they're seeing the southern black sea bass or the northern black sea bass?
I've never seen a northern black sea bass. I'm letting Kevin take that.
Yeah. It's been a while since I've had a good look at them that way. They definitely grow larger in that area. So I mean, other than that, I don't know. The blues pop a little more, I think, on the northern ones, but that's just anecdotal.
Do you know if there's a video survey up there where, you know, somebody could begin to look into this? Because you got to have a lot of videos to, or at least get AI to do it for you.
I don't know of one currently. I know there has been talk about doing some sort of video or video trap because of things like implementation of wind farms up there that, you know, trawl surveys can't get into those areas or work as effectively. And so whether or not to do some alternate type of sampling, but currently I'm not aware of one.
All right, and then Amy had another question for you all. Kevin, can you share what other associated data was provided with the fin clips that might be beneficial to use or identify next ideas for additional studies?
Yeah, we definitely have all the collection information. So the location that they were collected, the time, the gear, the size of the fish, that information's all there. I think anything else specific to that would be.
Yeah, and I think for the most part, these were ones that they were also planning on aging. If not, we probably can create an age link key to create those ages. Yeah, so it's
Yeah.
I mean, it's a very complete data set, so I'm sure we could do a fair amount with it.
Yeah, there's a routine, the CHESMAP and the NEMAP survey have a routine diet program also that likely the fish that they took those clips from, and they're also aging those fish as well.
Amy responded with yes. Haley, I see you. There you go. I'll unmute you. Go ahead, Haley.
Hello, can you hear me?
Yes, we can.
All right, perfect. So the statement I prepared was a little bit much for what type of seminar it was, so I'm just gonna kind of go over it. So my name is Haley Scott. I run a network of captains. It's basically a live feed to the inlet. So I have 15 captains in my network that range from 50 to 5 years in business. And I had a discussion with them this morning kind of about our local sea bass fishery because it's been in serious decline for the last at least 10 years. Just to kind of give you guys some on the water information and really no better information than some of these guys. So I'm just going to read pieces of it so I don't take up too much time. So it's nearly non-existent here. And while you guys up north are catching them, we don't catch any. We're lucky if we catch maybe 5 to 10 a year. It wasn't like this before the red snapper closure. We had a larger presence, but we don't actually blame it on the red snapper. It was more of the regulation changes surrounding red snapper that had the effect. So when the closures hit, now, before I say this, some of my captains are dual. So they're both charter fishermen and they're also commercial fishermen. And they said that once the closure happened in 2010, the commercial fleet here shifted to sea bass pops to make up for the lost revenue. And it worked briefly. And then the explosion of snapper competing in the same habitat and the pop pressure and the steadily warming water here just made the entire population here collapse. They stopped using the traps about 2 years, 3 years after the snapper closure because it's just the gear is too expensive and there's no point to do it when there aren't any black sea bass. I would say that the local stock here is overfished. It's not experiencing overfishing. But yeah, there's a lot of things outside of our hands that are environmental, ecological, that we can't really seem to help. And that's basically it. Thank you.
No problem. Thank you. And which area are you from, Haley? I'm sorry, I might have missed that. I know it's
Ponce
Florida.
Inlet. I might have skipped over it.
Thank you. And we definitely heard last week from the Snapper Group or AP that you know, there's not many black sea bass down there off of Florida now.
Yeah, no, there isn't none. Thank you guys. I appreciate it.
Thank you. Judd, I see you have your hand raised.
Yeah, I'll go ahead and ask another question. Thanks, Chip. Y'all probably already discussed it, but is there any plans to combine, you know, the indices from the northern surveys and the reef surveys, you know, through your vast modeling approaches, or any other way to get, you know, a coastwide abundance index?
Speak to my little heart, Judd. You do. Yeah, it would. We've definitely talked with NEMAP about that possibility. It would be amazing to do. The challenge is that the trawls have a very good estimate of the area swept in terms of effort. The traps, we don't know as well what the effective area sampled is to create that sort of, you know, continuous, you know, equally, you know, same unit across the whole coast right now. Folks have done some tagging studies with red snapper, gray triggerfish, a little bit of black sea bass to kind of come up with a pretty decent estimate of about 100 square meters. So, I mean, I think we could probably go forward with something, but that's definitely been on the radar as a possibility. And I think it could be done. I think we just probably need maybe a little bit more tagging specific to the bait plume in our traps on what distance they're sort of drawing fish from.
And similar work has been done on other species, right, to try to figure that out?
Yep. Yeah. So for triggerfish and red snapper, it's I think maximum sort of drawing is somewhere around 100 meters. So radius.
All right. The next question is from Carolyn Belcher. Can you speak to the potential of sperm limitation in black sea bass? I know older literature indicates it wasn't an issue, but is it possible that has changed?
Oh man, we don't have any. Oh, sorry, we don't have anyone on the repro team here. Where's Wally when you need him? Yeah. So protogynous hermaphrodites, the big ones are males. It seems that the reports we're getting from some of our favorite fishers in this area is there's only big ones left.
So Wally does have his hand raised if you want to pick on him.
Yes.
Go ahead, Wally.
He's doing a great job. That's what I was about to say the same thing. Tracy's doing the same thing. She had mentioned it's mostly just large fish that are there. So sperm limitation is actually the opposite of like what we deal with the grouper species, because we're only seeing the bigger ones, not the small recruits.
Thank you. All right, any other questions? I feel like we've gotten through most of them. Sorry, I'm reading the questions screen. All right, so synopsis of what we've heard today. That we're not certain if things are shifting.
We know within the region, they've moved to the north. We just don't know if that's past the current sort of stock boundary though. TBD. That means we come back for another seminar, Kevin.
Yeah, hopefully it won't be another 4 years before we get you all back. Should be shorter.
No, they we have the last, what, 50 or so, 50 to 100 samples that came in.
Yeah.
Yep. And all the data just came, we've got all the data in place. So everything's running, everything's good. The new PCR machine's been great. The new,
Yep.
yep. We just had a little scare of a government shutdown and access to the lab for a little bit, but it turned out okay.
I'm glad to hear the work is still going on. All right, I'm trying to pause to see if any other questions pop in my brain or others while we've got the brain trust up there.
You guys know where we are. You know, feel free to reach out. We can provide our email addresses. Happy to do so.
And Jack has his hand raised again. Go ahead, Jack.
I'll ask one more question and I'll leave you alone. The general consensus among the commercial fishermen, I think all fishermen, are that these fish have moved north because there's so many, an abundance of fish north of Hatteras. I was just wondering what you guys think about that.
I wouldn't be surprised. I mean, that's why we dedicated the time to get the genetic samples this past year. You know, because we had been hearing that from folks and folks have been asking the question, and we really felt it was a huge priority to figure whether or not that was actually happening.
Okay. Well, thank you for your work, because we're all patiently waiting to figure out what's going on.
Yeah, we wish we had better news and we had a way to do something about it, but maybe if we figure out some things on what's going on, then we'll have some answers.
All right, there's a question in the question box. Is this the only species of the is this the only species this kind of analysis has looked at, or is there a evidence of similar changes of center of distribution, changes in center of distribution in other species?
So we did the same sort of thing with red porgy a couple of years ago, and at that time, red porgy were really just concentrating into the center of the region and a little bit deeper rather than going to the north. But in other regions, there's been actually a fair amount of work in lots of other regions, Mid-Atlantic, Northeast, I want to say pollock and cod, Alaska. I used to work up in the Bering Sea, and we had huge differences in the arrowtooth flounder populations moving to the north from the Gulf of Alaska into the Bering Sea. Yeah, so a fair number.
But as far as species that span the entire East Coast, this is a relatively unique situation. There aren't very many other species. There's a lot of difference in sort of community structure at that Cape Hatteras break. You know, most things that are south of Hatteras are not found very often north of Hatteras. So this is one of the few species that is found all the way up and down the entire East Coast.
Triggerfish might be a good candidate for looking at this next because there are more reports, I think, in the last 5 years or so about them being in the mid-Atlantic.
Yeah, and then blue line tilefish and golden tilefish, other species, and maybe snowy grouper. And I think there's others online that have experience with vast modeling that might be able to speak to other species that might have similar or things that they worked on regarding that. Any other questions or comments about black sea bass? I think this has been a really good discussion. You know, there's a lot of concern with the species and whether or not it's going to that northern end. It was discussed I feel at length at the last snapper grouper advisory panel meeting. The council's been discussing it. So there's a lot of concern. If looking back to the presentation, Julie, when you had presented the distribution changes of black sea bass for the northern area, I was noticing, you know, a lot of the orange was no longer around Virginia. It was moving northward. And so was that the density of fish in those areas in the trawl survey?
Yeah, and I would have to go back and look again at the source of that, but I'm pretty sure that was the fishery independent areas where they were finding you know, high intensities of black sea bass.
And with their survey, is it done similar times to the survey in the South Atlantic region where it's mainly spring through fall? Not much work is done out there in the winter time?
Yeah, the fall, both have a spring, both the NEMAP program and the Northeast Bottom Trawl Survey have spring and fall components.
And so just looping Jack back into this conversation, Jack, do you think when the mixing would occur or going further north would occur, would that occur during the summertime, or do you think the northern stock would come down into the southern stock during the winter time?
You know, we don't know where these fish came from. You know, our fishing was always the best as soon as that water started cooling off in September, October, traditionally, and would stay really good fishing until about now, this time of year, when it started warming up. We never really talked about where the fish came from. We just knew that they would come inshore, and our best fishing off of Morehead City would be anywhere from about 50 to 80 feet of water was the primary depth we were. We didn't know where they came from. We didn't care because the fish were there. You know, we didn't talk about that a whole lot. I don't know. That's why I was asking you about that. In the folks south, that we don't catch a whole lot of red snapper here. We have decent red snapper fishing, but as you get into the southern states past North Carolina, you know, they're saying, and I meant to add this a while ago, that they think there's an interaction with the red snapper and the juvenile sea bass are also causing a decline in the stocks. And I had to say that on the fishermen's behalf because they really think that's what's going on below us. I'm sorry, I can't add any more information. That's all I can do. Thank you.
Yeah, and there might be some interesting seasonal components to the northern stock too, because I know one of the reasons, so NEMAP were able to collect fish in their spring survey in the mid-Atlantic states, but the Northeast Bottom Trawl Survey really had to wait until their fall survey before they really started seeing sea bass enough to really get the fin clips for the project. So there is a little bit of difference between where they're seeing them to when and where.
All right, any other questions for the trio? Well, I am not seeing any hands go up or any questions getting typed into the question log. So I want to thank you all for a great presentation. I really enjoyed talking about black sea bass today, even though it's not necessarily good news for everyone. So hopefully we can expect some of your findings to be released soon, and we'll get a chance to talk about those either later on this year or early next year. And with that, I do want to announce next month's seminar series. It's going to be talking about dolphin fish. And so that will be, let's see, May 13th. Brendan Rundy and Paul Rudershausen will be talking about 2 of their papers that they've published recently with some folks that are online that were co-authors of theirs. So, changing species and changing fishery management plans, but hopefully it will be a great presentation and we'll see everybody there. Thank you all. Have a good afternoon.
Thank you.
Thanks, everyone.